All right, we are live. Welcome, everybody. Thanks for joining us to the Fulcrop Science Podcast. We'll have a full intro, it's getting worked on and everything. But today, I wanted to introduce my friend Matthew Powers from Regenerative Soil Science. Out of all of the people that I know, he is probably one of the best educators and most passionate people in this space. So, welcome, Matt. I'm really thrilled to have you on the podcast today, and I can't wait for the interesting conversation that we're going to have.
Thank you for having me. It's great to see you. It's been a while.
Yeah, I think the last time we saw each other was at the Mushroom Festival here in Oklahoma City.
Yeah, okay, fungi. That was fun.
That was fun. So, today I wanted to talk a little bit about nutrient dynamics, specifically, carbon and how it interacts in soil. Because there's this controversy. It's something that I always hear from people who, grow with synthetics. They oftentimes say it doesn't matter if you're growing organically or with synthetic fertilizers because the plant doesn't know the difference between the ions that are either brought in through fertilizers, through conventional fertilizers, or through organic amendments and carbon-based fertilizers. So, what is your take on when that topic comes up? When people are saying, "Hey, the plants don't even matter, so it doesn't really matter." Do you think that's a correct line of thinking, or do you think that maybe they don't understand the full picture?
I would so the idea that plants are can't tell the difference, that belies that makes it muddy, because the reality is plants can't help themselves but uptake what's there, and they experience a difference depending on what we're going to do in the context. So I think the fact that it can't differentiate between ions and it just accepts them they have a different experience and it operates differently. So I would, what I said before we hopped on, I think that they have a misconception about the way the cycles work. And when you step out and look at the cycle, that's really where it all comes together for me. So I would just walk through it with them. Like, so you've got this salt, which is just a chemistry term for two things that are bound together that dissolve in water. Very great delivery service, but that it's the function of these things, not the things you're talking about. The microbes are doing chemistry, of course they're releasing ions, but it's in a completely different context. So, and it's in a different rate, it's in a different cycle, it comes in a different way. Uh, so from my mind, I would zoom out, and I would be looking at, when you do something that's alone, something that's a pure form, and you put it into soil, it's no longer pure. It's always going to have a context where it's going to bind, it's going to combine, it's going to react with the biology, and when it's a pure form, it's going to be a strong reaction. So, like with like synthetic nitrogen is going to do this thing where it's going to be lonely. It's going to destroy the soil organic matter because it's going to essentially be composting. Just like you need, there's carbon and nitrogen balance in your compost to get it to come to heat. You're putting synthetic nitrogen in there, imbalancing the soil organic matter and carbon to nitrogen ratios instantly, and then it's lonely. So it's going to rip things apart as it partners with things and forms a stabilization between those things. And in doing that, it's going to solubilize all of it and remove it. Biochar, when it's just char, you throw it on soils, it robs the humus, tears things apart, ruins your structure because it's lonely. So I see these things when we do salts as lonely, and so they're always going to have a negative effect. Microbes, they embody nutrients. They cycle and transform nutrients. They generate nutrients in some cases. Those things are naturally regulated. Those things are at a rate that plants evolved for, And we kind of see a proof of this. This is what we were talking about right before we hopped on. Restrepo Oliveira, that guy who wrote the ABCs of organic agriculture for Latin and South America. It's in Spanish. I think it may be in English now, but I only had it in Spanish. And so he basically said, well, what's in your shed is what's also stuck in your field and desiccating your organic matter and holding back your growth. It's like it's a clog. And so when you look at with the microscope, if you look at the desiccated organic matter, it's shriveled up. It looks like it's mineral, but you know it's organic matter from the color and the shape. And it has studs of phosphate crystals on them because they've been desiccated. And you know, hey, phosphate's one of the things that's here. He said, take all those extras, those little bits, the chemical fertilizers that no longer seem to work for you, put them into a ferment with EM, a rumen, molasses, rock dust, and ferment that, and then put that out on the field and have those microbes that are trained to eat that go and eat it in your field and finally cycle that out, unlock your fields so that you can start growing again. And so right there's your purified, simplified inputs, amendments, and chemistry going in, but it's fixing a problem created by the chemistry. You know what I mean? The chemicals that they added, the synthetic salts that they added. So it's not that they're evil and that salts are all evil. That's a thing. It's a thing.
Yeah.
It doesn't have a moral compass. It's on us to actually be we have to make choices and decisions that are ethical or you know what I mean, or better and better and better and improve over time, because we all live in the real world, right? So this idea that they can't tell the difference between ions hits me like in such a strange spot, because I'm like, well, there's all this huge context involved here, and then these things have different motivations and different pathways. I mean, exudation is going to happen regardless whether the plant's sick or not. If the plant's sick, it's going to release poor exudation and call in bad things to eat it and destroy it. It's the same thing with everything. The plant can't help it. It's really a passive instrument of photosynthesis. It's going to pump and do photosynthesis to the best of its ability until the day it dies. So I don't want to demonize the chemistry, the language of chemistry, or the actual elements or minerals. Nothing wrong with them. And it's the delivery, it's the way we're doing things. And so much of this comes back to what you mentioned when you invited me on, which is carbon. When we have these elements in the right forms with buffering of carbon involved, we remove antagonism, we remove the shock factor. And if we have the microbes doing this, they go into the plant as either endophytes, and then they release nitrogen and do other things, or as part of rhizophagy, and they get consumed, and whatever's inside them is released. So it's a completely different mechanism, but all plant roots do rhizophagy. All growing root tips do rhizophagy. And so this is part of the reason why things go in always, right? These roots are conducting ions, and these roots are attracting cations, you know what I mean, and anions, depending on what they need. They're always doing this. They're always absorbing. These roots are very sensitive organs of the plants. We got to treat them that way. And so so much of the salt chemistry is being leached away. So much of the salt residues are wreaking havoc in our lakes, rivers, and streams. But you don't have to spend all that money, and you don't have to be bound by that, ideology. They act like it's a religion, and that's really, I think, where we take issue. We're not like you could take your urea and combine it with lactic acid bacteria, yeast, and purple non-sulfur bacteria. In other words, EM.
Yeah.
And then
or microbe plus, which is
the same thing. Yeah. But you could also do water kefir, right? Yeah. Mm-hmm.
Or you could even add it to humic and fulvic acid. There's all different ways to create better efficiency with those things. when it comes to carbon dynamics, because you have, chelation to organic acids, which decrease precipitation reactions, which improves the overall chemistry of that system. And then you have microorganisms that might be producing organic acids and amino acids, which also chelate things. They might be producing sidiophores. So there's all of these different functions of carbon that interact with the chemistry. When we're talking about salt-based liberated ions, though, one of the things that I always like to think about is they're absorbed via mass flow. And in nature, there's three different ways that nutrients are acquired: mass flow, diffusion, and root inception. And so we're bypassing things like diffusion and root inception when we're just putting solutes in water, and the plant has no choice, because naturally the plant will diffuse things that it needs and doesn't need at specific times. And when you're using something and when you're using something that's completely soluble, the plant really doesn't have a choice but to uptake it, because it has to access that water.
And that's actually the original reason why NPK is like number one. They couldn't avoid these nutrients and these minerals, and like the atmosphere is over 70% nitrogen, right? You look at potassium, it's all the same form except for like a little tiny peak of the redox chart. It's like these things are ubiquitous, and their forms, like plants had to evolve to deal with them. And so I really see these things in a cycle, and I see the microbes doing it all. And so when we do salt, we're really causing a lot of problems. We're wasting a lot of money, and we're having to redo everything again because we just zapped our soil.
Yeah. So let's maybe talk a little bit about synganics, right? Because the combination of using, like you said, it's about being responsible and efficient. And so if we're taking a practical application to introducing nutrient ions that are soluble, but also complexing them with carbon and via microbial fixation through microbial mediated processes like the production of cytophores and organic acids, then we can improve the efficiency and the use efficiency of those salts and decrease things like leaching and runoff and the negative environmental impacts that those have, right?
Right. So your Bokashi ferment brew, what's that called again?
We have the Microbe Plus.
Yeah, the Microbe Plus. So that Microbe Plus, the plus on there is really key, because once it runs out of sugars in that brew, folks, it switches in succession to feeding on a byproduct from while they were feeding on sugars. Yeah. The byproduct becomes the food source for the next stage. Mm-hmm. The next stage starts, and what do they release? They release protons. So like H plus, the plus. And so that's what roots are doing. So then you can combine them with substances, interact and open things and do things as if you're in a root zone, so that when you deliver it to plants, it's already transformed. It's already in the form that allows them to be more absorbable. And so like chelating, like you're talking about, you could be using that brew in conjunction with bat guano or bird guano or something like that, or your rock dust or your kelp, and then you could be delivering it in a more bioavailable form, and you're really just imitating what the plant does, in a controlled environment, because those microbes in your brew right there are all also endophytes.
Yep, that's correct.
So we're taking the endophytes and exudate from plants essentially and putting them so we're pre-digesting, just like we do with so many things, with food, because it makes it more beneficial, more bioavailable, right? Our ferments, more bioavailable nutrition. And so we're doing that with a lot of these things. And you could do them wrong. You could take your hot compost, pH 8, because the most hot compost is pH 8, and then you could be putting it on there at the wrong time. We're gonna layer on a soil soak right as we're flowering, right? And it's like all nitrates, and then you could have a fungal just bloom. And so yeah, we can do things wrong with natural things too. and we can do things right with these substances. But when we use the salt, we have to be very careful. We have to have a plan, and it has to be partnered with biology and carbon, because if you just have no carbon and you add it, you'll kill the microbes.
Yep. Yeah, high salinity situations, some microbes may survive, but it opens up the potential for pathogens because you're depleting a lot of the beneficials, and it's a lot of times due to the osmotic stress that's because those microbes also have to take up soluble nutrients from their environment and water, and if that water contains high salt concentrations, those cells can lyse and then go through apoptosis,
Right.
and we don't want.
It's like
Yeah.
the biochar. They're all needy. They're lonely. These isolated ions. They're like, they want to bond with everything, and they tear everything apart in the process.
Yeah. And you know what's really cool about the fermentation process too? Because what's happening is they first consume a carbohydrate, and things like the Saccharomyces, which is naturally generating CO2, is then consumed by the Pseudomonas, the photosynthetic bacteria, and that photosynthetic bacteria is then producing an amino acid that can be utilized and transformed by this Bacillus species. And so it's like a cycling of an engine. There's an internal cycle that's happening with the metabolites that are being created amongst the consortiums themselves. And then once all of those things are consumed, what you're left is not just the microbes in the bottle, but you're left with all of the organic acids, all of the exudates that those microbes made, all of the amino acids, the sidiophores, those phytohormones that they were producing are actually captured in that process. So then when you're putting them into a system, you're not just getting the benefits of the microbial inoculation, you're getting the benefits of all of the compounds that they produced during that process.
Absolutely. And if you keep it going through those successions of feeding, once it releases, I mean, you'll literally see things expand and then suddenly contract. And you'll be like, whoa, did it die? No. It's consuming that now. And so it's that CO2 is being reconsumed and turned back into a different form of carbon, and it's wild.
It really is.
They're using it as a tool. Their byproduct, they're like, oh, now I got nothing but this byproduct. Switch gears. And provenance of a bacteria, I always say it feeds 4 different ways. Yeah, but within each of those 4 different ways, they have modalities.
Yep.
So they have a lot more than 4 ways to feed. They have 4 main ways that they feed, like photosynthesis, consumption of organic matter,
Yeah.
But
there's 4 modes of metabolism that they can cycle through, and it's absolutely amazing because whether it's fermentation, whether it's photosynthesis, whether it's degrading organic compounds or using inorganic compounds, so that it's
they can bombard other microbes with energy in order for the other microbes to die and release what they want.
Yeah, it's pretty interesting. It's pretty interesting how they work. And so when we're looking at, these different cycles in soil, yes, ions, an ion is an ion, right? But it's the chemistry behind that ion. If we take something like the phosphate ion, which has a negative charge, we know that depending on pH, those things could bind with calcium and to a lower extent magnesium at higher pHs and micronutrients at lower pHs. But when you have that carbon, right, that fuels the microbial processes, you decrease those precipitation reactions and create higher efficiency.
I think a big part of that is also differentiating which soil organic matter you're talking about. I was just literally just on a podcast an hour ago, and I was talking about how if we put this, and this is all dry, this is an IOMO 5, and then this is 4 years old. It's been in this bag for 4 years, and it's still wet. It's still amazing, and if we put this in a test tube and shake it up, there'll be all this turbidity. It'll be chocolate brown and opaque. Can't see through it. If I do it with this will immediately settle to the bottom. It'll be like cloudy water, but I can see through it, and it'll be like whitish cloudy, not chocolate brown. And it'll quickly clear and and this will be suspended as chocolate for days. And this is nothing but compost. This is not like fine clay or silt that's being suspended. this is organic matter that's so fine because it's been recomposted. It's Johnson stew that's been recomposted. So that we got to remember, there's a difference between soil organic matter is not all the same.
Yeah.
And that small, super fine humic compound and humates and all these different, like your product, this literally is so stimulating to microbes because it's so tiny that it's so energetic. And think about being surrounded in a world where all the interactions have like a maglev like buffer on them, so it creates smooth movement. It's literally not just a buffer that where you squish on our between our sand, silt, and clay. It is that, but when it gets super fine, it actually creates a force field around it that creates buffering that's energetic. And so these microbes can zip around. These microbes, it's better movement. It's better for the economy. So the exchange of nutrients, the release of nutrients, and the holding of water, the holding of colloids. That's how colloids form. People talk about easy water. You know, when I talked to Dr. Gerald Pollack, I literally was like, so if I put alginate in water and shake it up and put it in a blender, that's your easy water, right? That's the fourth phase water. And he was like, well, yeah. And I'm like, we all can do it. And in fact, it's like very, very natural for these things to happen. It's colloids, you know. He just gave the term, the formation of it a name. And so we can do this, and we can see it form, and those colloids create a crystalline structure that's energetically charged, and microbes move through this, nutrients move through this, nutrients are conducted through this in ways that nature intended. This is what nature does when it's allowed to thrive and grow, and we can do that in our soils. We can work our soils and make the earth for our plants as regenerative as possible. But salts, you know, just applying salts on there, I mean, it's like, we got to do surgery. Let's shoot a hole open in the person and then, you know, go in and get that organ. It's like, you don't need to shoot a hole in the person. You can use like a knife and like, you know, sew them up nicely and stuff. It's such a waste of energy, time, effort, and your money.
And resource too, because when we're thinking about, you know, the logistics that go into the production of fertilizers, we're looking at either a massive amount of energy being produced that's very efficient to create nitrogen from the Haber-Bosch process.
Right.
And then when we're talking about phosphate, we're talking about mining, all the fuel that goes into that, the conversion, the micronization of that, and then they also have to have a sulfuric acid plant, so they have to manufacture sulfuric acid to attack that rock phosphate to liberate the phosphate to make phosphate fertilizers, which are then combined with the ammonium that's produced through the Haber-Bosch process to make things like DAP and MAP. And just to make something like urea, for instance, to make 1 metric ton of urea, it takes 66 million BTUs. That is a massive, massive amount of energy. And then at its greatest, urea only has probably about a 30% use efficiency. So it's recycled back into atmospheric gases, or it's converted into nitrate, which doesn't hold on to soil colloids that don't have enough organic matter, and they end up leaching out into our waterways. And so we have all these logistical inefficiencies when it comes to the management of the resource, the way it's produced, and then the way it actually is used and how efficient that product is at the end.
I feel like so many of them are just being duped. I mean, someone who's like, oh, I need my phosphate. I'm like, really? You need to pay someone all that money for no reason. Yeah. The reality is, it's like arbuscular mycorrhizal fungi, once it's established, is regenerative. So it'll come back every season, or it'll be in those grow bags if you manage your soil properly. Mm-hmm. The reality is, it can provide off the bat, like in some cases, all the phosphate you need. Yeah. And if you need more, like if you're a particular crop or a particular soil, all you would do is a foliar spray, and you would do it when you're fruiting or in reproductive. and that would be appropriate. You know what I mean? And you could do a small amount, and you could probably do a localized. I mean, there's so much phosphate in urine, it's crazy. That's originally where we discovered phosphate, right? So we have the ability to generate this stuff without mining, and it's a $72 billion industry. So remember that when you ask Grok or ChatGPT the question, it's going to do the same thing that it does when you put up pictures of certain people and it says the wrong. It says, "Oh, that's Jeff Bridges, not Jeff." You know, it's the same thing. If you say like, "Does phosphate glow in epifluorescence?" It'll go, "No, it can't do that." Wiki will be like, "No, it can't do that." And then you better believe you take some triple phosphate or any form of phosphate I can find, you put it under this thing, and it'll glow for you. So why would they be against an easy way to verify if your soils are choked by phosphate? Because you can see the little crystals, or that your roots have mycorrhizal fungi and you don't need to buy phosphate.
Yeah.
Or
there's another instance too, because phosphorus is an abundant mineral, but it's just in a low quantity. So a lot of times what you need to do is you need to increase the carbon dynamics of those soils, so that way the phosphorus cycle works more efficiently. So adding carbon to the soils and then inoculating again, whether it's mycorrhizal fungus, whether it's a consortium like the Microbe Plus or EM, that is going to liberate and the parent apatite material and convert it into phosphate.
And that's carbonic acids, but also energetically, that is literally that conveyor belt of the maglev relationship of the formation of colloids, the formation of soil organic matter pathways and structure.
Yeah.
So it's really wild. We live in a world where a lot of people are duped. A lot of people are just adamant about this religion that they've been fed by a group of people who have not their best interest in mind. So my work really has been to ask, how does this happen in nature? What's most natural here? And then, what are farmers doing? What are growers doing? And how can we connect them to the cycles and then have them see it and go, "Oh, that was stupid," and then do something better because it's obvious to them. And so that's really what I try to lead with, because so many of them just literally are just being taken advantage of.
Yeah, and I think I read somewhere it was like a 0.1% increase in soil organic matter can equate to like 20,000 gallons of water holding capacity. Yeah. So it's like not carbon doesn't just act as an energy source, but it acts in so many different chemical dynamics with nutrients and soils and how those things perform, along with things like the nitrogen cycle, the carbon cycle, the phosphorus cycle, micronutrient cycling, and then also the microbial cycles that naturally happen in soils. So it's like mind-blowing how complex these systems are, and I don't think...
And efficient.
Nature always has to take the path of least resistance. That's the thing, because in nature, that's the way that physics works. If you can get somewhere in the quickest way, that's the path. The path of least resistance will always be taken in nature, and so it's built these really, really efficient systems that these big companies thought, oh, we can bypass this, we can do it better. But it's like it's not really making it better. Well, you might see instant results because of the delivery method, the logistics behind it, and the long-term impacts are all negative.
The Brazilian rainforest is a great example. They're basically taking a system that they don't understand because they say it has poor soil. Well, it's actually hyper efficient. The reason that the soil there's so many plants and there's a successful rainforest there is microbes.
Yeah.
And so without those hyper efficient microbes that are nitrogen fixers and endophytes, transforming the detritus as fast as possible into nutrition, and then also, working with the rain, it wouldn't exist.
Yeah.
And so now what do? They chop it all down, and then they do synthetic, chemical salt ag, and the entire landscape lasts a few years and then just turns into desert. And so it's this paradigm, and when people see it, they're like, yeah, how is that possible? And they're like, that's the question. How is that tree growing out of that rock on the side of the cliff. It's like there are things going on in nature that defy all the university ag, paradigms. And we just have to leverage those, and we'll see such an incredible difference.
I absolutely agree. Well, we're coming up to the 30-minute mark. I don't want to keep you too much longer. Maybe we could take a little bit of time to do a little Q&A on the chat. I think we got about 70 people in here.
Oh, sweet.
Going back and forth. So if you guys have any questions for myself or for Matt, just go ahead and drop those in the comments, and we'll do our best to answer your questions for about the next maybe 5-10 minutes, depending on how many questions you got, and then we'll sign off.
All right. I've got a course coming up September 15th, Regenerative Soil Microscopy. We'll be diving deep into all these things I was mentioning and talking about, so you can learn how to evaluate these things yourself, and you can help farmers get through that transition, because all farmers have to do is respect their arbuscular mycorrhizal fungi, and it will always be there. And it's a 30 to 40% gain in yield. So it's a no-brainer. So come get training with me, regenerativesoilmicroscopy.com, and we'll dig deep together.
Yeah, and if you guys haven't had a chance to check out Matt's books, I highly, highly recommend them. They're great reads. They're full of tons of information. Regenerative Soil Microscopy. And what was the other one? Regenerative Soil Science. And so those are great books. They're great references. If you guys aren't familiar, go check those out. You can find those on your website as well, right?
Yes. You can go to the permaculturestudent.com and buy the books there. You can go to regenerativesoilscience.com, or you can go to regenerativesoilmicroscopy.com and check out the new course.
We actually do have a new mycorrhizal. It's what was formerly Glomus intraradices, but Rhizophagus irregularis is now what it's called. But it's going to be pure spores, just like all of our our trichoderma and bacillus, and it has 30. I think it's 3.2 times 10 to the 8th power. I think it's 32 million CFU per gram. So nice high CFU in there as well.
I love it. Yeah, irregularis, the crazy thing about it, it may be the irregular one, but guess what? It's the dominant one and the most ubiquitous. So no matter your climate, no matter your soil type, you're going to find that arbuscular mycorrhizal fungi in your environment, in your natural environment. So yeah, it's kind of an incredible thing. But in nature, there seems to always be this Pareto distribution of hierarchy. So it's amazing.
I've come to the conclusion that most beneficial microbes will not outcompete each other, given that they have the proper resource, right? As long as there's enough carbon, there's enough nutrients, enough water. It's when there are limitations on any type of resource that they'll start competing.
Yeah. And what I meant by that was not like they're like push each other out. What I meant was like their actual amount of them. So there's like tons of E. coli, and then like tons of Streptomyces, and then less like Burkholderia, and then less like it'll go down in a hierarchy. So there'll be like a top 1%, and then 0.1%, and then 0.01%, and it goes like 2, 20, 50, 100, and then it's thousands at 0.00001. So it's like it's very interesting because you see the same microbes in the top 25 every time.
Yeah,
that's what I meant.
Really interesting. So we have a question. It says, not directly related, water testing. Is water testing like soil testing, where you want to find a lab and stick with it, because like soil testing, each lab has their own set of SOPs?
I think it's really good to know the different SOPs, because then it's like how the sausage is made, because the reality is you're going to want to know what that nuance is. Like a Haney versus an M3, the Haney is imitating exactly what plant roots release, right? So that's like a much more accurate test. I mean, I don't know of any company. I have students that are doing testing.
I do testing. I do testing and I use Logan Labs exclusively until this new soil lab is built that my friends work at. But I use the same lab, just like if I was going to do soil testing, I do it for water testing. That way, you can learn to read their data, and because the data is going to change, and if the data changes, you don't know how to interpret it. So using the same lab, in my opinion, is the best approach.
No, I totally agree with that. I was just trying to say that like I tend to do multiple tests because they all have different strengths and weaknesses, and so I'm also doing it for my students and my books and my research. So I do extra tests, but
Yeah.
yeah, on a regular basis, I'm using a microscope to do tests, and I do that at home.
Which brings us up to our next question here. What is the best way to get started in the microscope usage? He says he works in the medical field and have a top-notch microscope, but have no idea where to begin.
Well, you're going to want to get a book or something that has microbial profiles and directions on the protocols on how to sample and then view your compost and soil. And that's what this is. This is the first book that ever did that. So this will tell you how to use the microscope, the way we do it, what microscope types, how to operate them, how to do the protocols, how to do the drops, how to do the ratios, and then how to interpret it. And then case studies and microbial profiles are also in there.
Awesome.
And then the actions, that's the first book, Regenerative Soil. So they work together. But that's what I would do. You've already got the microscope, you already got the skill. It'll be easy for you. The biggest hurdle is developing your eye. So in my course, the first 10 weeks is morphology. We're going over, and I'm breaking a lot of myths around morphology. People think that nematode mouthparts are static. They can be dimorphic. They can change in response to the food that is there. So the soil food web concept around that,
that's really interesting.
Yeah, there's a lot of that. So I break a lot. So much of it is myth busting, and so many of my students are from soil food web school. So I have to go through all that and show it inside and out. And then the second half, once your eye is ready and your paradigms for everything are ready, I model it at long scale, talking the whole way on video, and then I do it quickly live. And I use my microscope's camera hooked up to Zoom, and you guys see it in 1080 HD. And then I have you guys break up into groups, and then you plug because I teach folks how to do this, you plug your microscope camera into Zoom, and then you take your small group, 3 to 4 people, on a tour of your drop from your compost or your soil, your biofertilizer, your vermicompost. We have themes for each week. So you end up seeing 12 to 14 samples of hot compost. And that develops your eye better than anything else can, because you're there with a group of people that you've been meeting with for 10 weeks straight. You know them. You've been doing this group work in preparation and planning, so you get all the right equipment. You end up with authentic learning, social learning, real-time learning, and you get all your questions answered in real time. And I can actually look at what you're looking at and be like, oh, that's that. And so there's nothing else like it out there. And so I really highly recommend checking out Regenerative Soil Microscopy, the online course, if you're interested in this, because that's how you develop your eye in real time with other people with real samples.
Awesome. That's great. That's a great way for people to learn. Last question is, what makes your microbe plus better than the competition? I would say that the CFU count is higher. So the higher amount of colony forming units, the better they're going to be able to colonize your substrate.
And that's going to be something you can even tailor. Like you could take his brew, you could be extending it, activating it. I don't know what word you guys are using, but when you do that, I pour in purple non-sulfur bacteria extra. And then I'm putting in water kefir. The water kefir is not added to these things. These guys are doing it from spores. They're doing from pure cultures, which means that they're deciding the ratios and the amounts. And so I look at mine under the microscope and evaluate it, and then I'm like, I'm going to add more of this, And
That's
so Yeah, it's a lot of fun, and it just shows you how powerful you can be with your choices. You can verify things. You can see how awesome Brandon's stuff is. You can check it out. Mine is actually. I have a little bit left in mine. Mine is by the sink out there. I should have grabbed it, but I have the Microbes Plus 2, so it's a wonderful product. And these products, they really, really work. I think a lot of people are like microbes, bugs in a jug, but just try it and just try one extension and see the difference. And also, the other thing to think about is if you've been building your living soil for a while, you should feed the microbes something specific with a goal in mind so that it does that for you. A lot of people who've built their soil, they've got all these things, and yeah, EMs, they're naturally, or Microbe Plus, these microbes are naturally endophytic.
Yep.
So they're already going to be in a small scale in your plants. So you got to do enough. You got to have enough there, and sometimes you got to feed them the right things so that they do the work for you in a way that you can see.
Absolutely. Well, Matt, this was a pleasure speaking with you. Tons of great information. If you want to give a shout out one more time where people can find links to your books, your courses, and everything else, and then we'll sign off.
RegenerativeSoilScience.com, RegenerativeSoilMicroscopy.com. Come visit me, come check it out, and come get a book. I'll see you soon.
Awesome. I'll see you everybody later. Thanks for joining. Until next time, everyone.